Okay, welcome everyone. It's great to see so many familiar faces on the call and a few new ones. Today we're going to be hearing from Brendan Reardon, who's going to be talking to us about the Molecular Oncology Almanac. It's a precision oncology resource hosted at the DFCI. Brendan's going to give us a walkthrough of it, and there'll be some time afterwards to have a little bit of discussion about the resource. So without any further ado, I'm just going to hand over to Brendan, who can give us a walkthrough of the Molecular Oncology Almanac. Take it away.
Great. Thank you so much, Alex. Let me share screen. Can everyone see my slides okay? Thumbs up?
Yes.
Great. So as Alex very kindly introduced me, my name is Brendan Reardon. I'm a computational scientist in the VICC lab at Dana-Farber Cancer Institute. I'm also a doctoral student at the University of Limerick. I really appreciate you inviting me here. I started coming to these this talk series, I think around December or so, and I've really enjoyed it. So it's a big honor to present here today. And I really appreciate you all coming, especially with AACR ongoing at the same time. As Alex said, I'll be talking about computationally aided clinical interpretation with the Molecular Oncology Almanac, which is a paired clinical interpretation algorithm and underlying database for precision cancer medicine. The slides take me about 40 minutes to get through, so there should be ample time for Q&A and also give everyone plenty of time to get to their next meeting and or presentation. So the structure of this presentation is kind of giving an introduction first. It's not needed as much for this audience, but I thought I would just go through it as anyway. So what is computationally aided clinical interpretation for precision cancer medicine? Next, I'll introduce this resource and showcase some features and observations that we've made by applying it. And then lastly, I'll end with a brief bit about limitations and future directions of the resource. So starting out, very generally, and before I start as well, I do have the Zoom chat open, so feel free to, when I'll return to this structure slide as the presentation progresses, and that's a really great time to kind of pause for questions, but feel free to turn on your mic and ask questions throughout it, or type in the chat. I do have that open on the screen in front of me, so I'll be able to see. So anyway, what is computationally aided clinical interpretation for precision cancer medicine? Starting very generally, we each have 4 to 5 million variants in our inherited genome, which differentiate us from one another. In the case of cancer, we can develop multitudes additional somatic mutations. And a promise of precision cancer medicine is for cancer treatments to be tailored to individuals based on the genetic changes within their tumor and their inherited profile. So very broadly, in the case of advanced cancer, we can test for tumor-specific biomarkers that can be drugged that result in clinical action. For example, EGFR, mutant EGFR, or erlotinib, BCR-ABL, imatinib, et cetera, et cetera. And kind of, thinking about how this is implemented, we all know this audience where bench-to-bedside leads research goes on to impact physician recommendation to guide individual patient care. But in this audience, we know that it's a bit more complicated than that, where we have a bunch of prior patients who are enrolled in various clinical trials and research studies, and these studies are going on to produce papers that are published in the academic literature. These will go on to be expertly curated into various different data sources, such as the clinical guidelines from various organizations, such as the NCCN, that guide clinical practice, or even the FDA for specific approvals. And then these resources are used to actually guide physician recommendation for new patients. But there's a lot of points of friction here and challenges. One, technology at the point of care is rapidly evolving. Most academic medical centers, we have primarily DNA-based targeted panels, but more and more we're seeing patients receiving some transcriptome sequencing. We have single cell, which is blown up seemingly over COVID, immune assays, multiplex IHC. Whole genomes are becoming more and more affordable, never mind proteomics and several other data modalities that we can use to characterize the genome, the data points per patient. Furthermore, knowledge keeps evolving. Every day there's a new scientific paper coming out, and it's a lot for even us as researchers to keep up with, never mind physicians who are also seeing patients. And thirdly, to that end, clinicians generally need help. with kind of making all this information digestible and figuring out how to actually take these genomic alterations and associated clinical actions and figure out which ones to prioritize and kind of just generally help the molecular tumor board interpret this information. So there's an idea. Maybe we can use the power of computer science to help with all this, and maybe we can add clinical interpretation systems to help with this process. And again, I don't have to as much with this audience, but just to be very clear with what our goals are, we can kind of think about 3 models for interpretation in cancer research. So on the left-hand side, we kind of have the status quo, where molecular pathologists review patients and make decisions solely based on clinical guidelines and their collective knowledge. In the middle, we have a model that molecular pathologists review patients and make decisions with clinical guidelines, their collective knowledge, and computational systems to aid in this process of knowledge acquisition and profile contextualization. And then thirdly, on the right, we kind of have a more extreme circumstance where you can imagine a computationally driven molecular tumor board or trial where a computational system will just rank and provide recommendations for patient care, and the pathologist might just kind of checks off and sanity checks the recommendation. This is not what we're talking about. We're really trying to work towards and add value to a future where different algorithms, software, and databases can greatly help with the sample contextualization and knowledge acquisition. For example, wrangling the latest and greatest research, presenting it in a digestible manner. Physicians and molecular pathologists are busy enough as it is, and such systems might be able to provide aids to give them additional information and help them do their job as best as efficiently as possible. So we created a paired algorithm and database called the Molecular Oncology Almanac. I'll tell you more about this in a moment. We had a publication that came out in late 2021. Time flies. But before kind of going into the resource and providing some observations from findings, any questions or comments so far? Great. Okay, so I'll move on to the next section. Again, feel free to pipe in at any time. So we created this paired algorithm and underlying database that we end up coining the Molecular Oncology Almanac. The way the algorithm works is it takes in any combination of somatic variants and germline variants from the patient's DNA, as well as copy number alterations, and as well as somatic variants or fusions from RNA. We'll annotate and evaluate these, we call the first term genomic alterations, so just kind of individually looking at them with several established databases, as well as our underlying knowledge base. These several other knowledge bases are by proxy doing a biological or functional interpretation for oncogenicity. We can talk more about this later. And so we'll evaluate with several other data sources, specifically cancer hotspots, 3D hotspots, cancer gene census, COSMIC, MSIG-DB pathways and modules, as well as EXAC and ClinVar. And then our underlying database is what's used for clinical interpretation. At the moment, we have nearly 900 citation-driven clinical action genomic alteration relationships that assert therapeutic sensitivity, resistance, or disease prognosis. So again, first we'll look at all genomic alterations that are provided on a 1-by-1 basis. Then the algorithm will kind of go through and do a second pass. We'll look at integrated features, so sequencing concordance, as well as germline somatic overlap, for example. We'll also look at genome-wide molecular features, such as microsatellite stability and aneuploidy, as well as computationally inferred molecular features, such as mutational burden and mutational signatures. Last but not least, we also have a very much for research use only module where cancer cell lines from the CCLE that were deemed genomically similar to the provided molecular profile will be reported, and then the shared genomic features to the provided molecular profile and the drugs that these cell lines are sensitive to can be reported as well. Many of these different features I described in this workflow diagram can be turned on and off with the configuration for the algorithm, but all of this will be included in the patient actionability report, which we'll talk more about in a bit as well. So the way the algorithm actually works to do the clinical interpretation is kind of a three-step process. So when evaluating individual genomic alterations, first thing we'll do is we'll match by data type and by gene. So on the left-hand side, you kind of have a cartoon schematic of presuming a patient with non-small cell lung cancer harboring a EGFR T790M variant. Very well characterized biomarker. And first thing the algorithm will do is look in the database and split out the database by data type and grab the EGFR somatic variants. Next, it's going to match by ontology and additional feature details. So in the case of somatic variants, that's the variant consequence, the protein change, for example. And you see in the cartoon, we've split up the database entries in EGFR by non-small cell lung cancer and then other tumor types. And then thirdly, we're going to select the strongest evidence, and then additional matches are returned as well in the report. And so here we're going to be returning the obviously FDA-approved biomarker for T790M as kind of the top results, and then additional matches will be returned as well. This curation of the underlying database happens a few different ways, and this is described in our standard operating procedure, which is on GitHub. So FDA approvals and clinical guidelines are added to the database after internal lab review, just because those are kind of the most safest information. We want to try to keep on top of that as quick as possible. Academic literature is amassed with the help of medical oncologists or domain experts, and then vetted by both them and internal review. And then periodically, we'll perform cancer type specific reviews with medical oncologists or cancer type experts to gather feedback, both on assertions and records that we've had in the database within their domain of expertise, and as well as the project as a whole. And so if we look at our underlying database, as I said, we have 874 relationships at the moment. These are from a variety of different evidence sources, in addition to FDA approvals and clinical guidelines from the NCCN. We also derive relationships from published academic literature about clinical trials, results from clinical trials, clinical evidence, so studies in humans, preclinical evidence, so mice or cell lines, as well as computationally inferred findings, such as like mutational signatures or something from clustering, for example. And all of this is kind of in service to extend the long tail of actionability for patients who might be variant negative for a well-characterized biomarker. And one of the things we did is we looked from AACR Genie, where, for those of you who don't know, Genie was is a effort to kind of share data across different medical institutions. They just had their latest release of, I think, 100,000 patients within the past few months, which is really amazing. And it started out with, I think, 18 institutions back in 2018 or so. But Dana-Farber was one of those first institutions. So we looked at patients who received targeted sequencing with OncoPanel from Dana-Farber, and we annotated them with our algorithm. And then in this figure on the right-hand side, we kind of have a box and whisker plot where every data point in the box and whisker plot is a different cancer type. And on the x-axis, we have considering levels of evidence cumulatively. So the far left one is only considering biomarkers associated with FDA approvals. And then moving one right over is including biomarkers associated with clinical guidelines. And on the y-axis, we have the percentage of patients within that cancer type that did have a genomic alteration associated with the biomarker. And again, it's cumulative. So, as we move right, we're considering more and more evidence. And what we see is that by considering FDA approvals and clinical guidelines, we had about 27% of patients had at least one genomic alteration associated with therapeutic sensitivity. And if we look here, there are some cancer types that are pretty high, where most patients do have a genomic alteration in them. And those two data points at the top for both box and whisker plots are largely dominated by KIT somatic variants in gastrointestinal stromal tumors, which are associated with imatinib and other similar FDA-approved therapies. But if we look, we can see kind of median about 12% of patients across different cancer types did have a biomarker. And if we include additional evidence sources that we've cataloged, we're thankfully seeing that additional patients are receiving a therapeutic hypothesis when they were otherwise variant negative. This is represented as we include additional data types, so including clinical trial evidence, preclinical evidence, and inferential evidence. The cancer types overall are moving up on that y-axis, where more patients are receiving or being associated with genomic alteration associated with a drug related to therapeutic sensitivity. And by looking at up to preclinical evidence, we're seeing that 27% is going up to 55% of patients in the oncopanel cohort were at least able to provide at least one therapeutic hypothesis to. But it's not just levels of evidence that we're trying to use to extend the long tail of actionability. We're also looking at various, trying to be inclusive of data types as much as possible. So on the right-hand side, we have another cartoon and figure that from our publication, where we looked at 59 patients with osteosarcoma, pediatric patients, and genomic alterations associated with different feature types. In this figure, these 59 patients are vertically aligned. So between somatic variants and copy number alterations, those 59 patients are lined up vertically. And in orange, in this figure in particular, we're comparing to a prior method from the lab versus the Molecular Oncology Almanac. In orange is that patients who had a clinically relevant event from Molecular Oncology Almanac and the lab's prior method. And in blue is patients who only had a genomic alteration observed from the Molecular Oncology Almanac. In addition to somatic variants, copy number alterations, as I said before, the algorithm will look at rearrangements, germline variants, aneuploidy, tumor mutational burden, mutational signatures, as well as MSI. And even though, a lot of targeted panels at academic medical centers are primarily looking at somatic variants, copy number, and select rearrangements, in this case, in osteosarcoma, we're seeing many other hits, clinically relevant hits in different feature types. And so we reckon more data per patient as possible is going to give us a more complete view of the patient and may even provide some additional therapeutic hypotheses. We've also worked really hard with various different scientists and partners to kind of grab precision oncology relationships beyond just targeted therapies and immunotherapy, which we mostly think of when we think of precision oncology. There's also a lot of really exciting research finding genomic associations with response to chemotherapy, radiation, hormonal therapy, and even combination therapies. And more literature is coming out, so we're really excited to see how the space grows. We've also worked really hard to make all this really accessible. In addition, so we have a website, MOAlmanac.org, where you can browse the knowledge base. This also links to our GitHub, where you can access the algorithm, all the code from our study, and figure generation, our underlying database as well. And everything's under a permissive open license. The database is also programmatically accessible through the API. I mentioned that the algorithm produces a patient actionability report. And we've iterated with collaborators early on to make this as simple as possible. So they're interactive web-based reports that are less than a megabyte in total, and all the files and pictures and information are self-contained, so you can send the report over Slack or email if you want to, and it'll be fully self-contained. Here we see some metadata and the generation date for the molecular profile provided. And then it's broken up into sections for therapeutic sensitivity. Here we can see on the left-hand side, or in the center, we have the observed genomic alteration from the patient or molecular profile. On the left-hand side, we have kind of the evidence that it was matched to, as well as how close the genomic match was. And on the right-hand side, we have the therapy and rationale, as well as source that was matched. You can also click on more details on the left for additional matches. We also do this with therapeutic resistance and disease prognosis. We also made a web-based portal on top of the Broad Institute's Terra platform, which is just built on top of Google Cloud. One of the things we were thinking about is that our audience is really molecular pathologists, initiated physicians and researchers, but not everyone's going to know how to use GitHub or Python, for example. And while a lot of the world is being connected through APIs, one of our values, and mostly both value and also requirements of some of our clinical partners, is that CLIA environments, at least at the Broad and Dana-Farber, aren't allowed to be connected to live APIs. They wanted everything to be packaged. And so we packaged like the whole algorithm and dependencies into Docker containers. And so we built a cloud-based portal on top of Terra in Google Cloud. So that way you can execute, like kind of upload data and execute the algorithm on your cases on a one-by-one throughput case, and then view your previous analyses, open up the workspace, open up the report. One trade-off here is that we didn't produce a web portal where you can kind of just input it and hit go and it'd run on one of our servers. It was really important to us that we wouldn't see anyone's data. And so you do have to kind of have your own Google Cloud billing set up. They did, and I believe they still do offer like free credits and things like that. But that was a trade-off we made where we did not want to see other people's data from a privacy point of view. We worked with Dana-Farber Legal and such to make sure that was all kosher. An additional thing is that you may notice that from this slide that this very much is kind of a pipeline where we're indirectly leveraging molecular tumor data from prior patients to inform what to do about new patients. And we're wondering if there's any way we can also directly leverage molecular tumor data from prior patients. And those would be fed back into the clinical interpretation system. So we also worked on similarity models to compare the patient to cancer cell lines. I think this was actually inspired by maybe AACR in like 2018 or so, actually, where I remember going to a talk and someone presented like a big PCA plot and said, you know, in 20, 30 years, we're going to put everyone on all our patients on Partners Healthcare into a big PCA and do nearest neighbors. That's really far time away. We wondered, well, is there anything we can do to kind of take a step towards that future? And again, in service of extending the long tail of actionability, where maybe a patient is variant negative for an approved biomarker, but maybe we can look at their genomic similarity to other patients in a population. And in absence of large, robustly clinically annotated patient cohorts that are available, we decided to start with cancer cell lines. And so kind of the spirit of it was, for a given case profile, look at comparison profiles. And we decided to sort first on the basis of clinically relevant features, and then further differentiate with a larger gene set. After a lot of different model tinkering, we end up evaluating with some metrics from information retrieval, where we kind of frame the problem as a sort, kind of, where we're given a case molecular profile, you're kind of querying comparison profiles and for which ones are relevant or not relevant. We decided to evaluate relevant neighbors on the basis of if a therapeutic sensitivity is shared. Prior approaches that have been published have defined genomic similarity on the basis of kind of an average bladder cancer in TCGA, for example, or kind of expected genomic events according to paradigm for like lung cancer, bladder cancer. But we wanted to have this be clinical actionability focused, so we used shared therapeutic sensitivity. And so on the left-hand side, you see a cartoon where you have a case profile, and then the circles are representative of cell lines, yellow if they share drug sensitivity, or gray if they do not. We evaluated this with a hold one out approach. So we'd hold out a cancer cell line, run different algorithms to sort them, and then evaluate. And then less similar, more similar. We ended up trying a bunch of different models that were relatively interpretable. Where on the right-hand side, you kind of see a scatter plot that was included in our study showing the performance of these various different similarity models that we tried. And in short, on the x-axis, we have the nearest neighbor average precision at the first neighbor. This is essentially how often is the nearest neighbor also sensitive to the same drug. And on the y-axis, you have the mean average precision. And that's essentially how good is the model at putting relevant neighbors at lower ranks or close, being deemed more similar to the case profile. Again, these are metrics from information retrieval. So you can imagine a Google search or something like that. And what you can see is that our x-axis is how often is the nearest neighbor also sensitive to the same drug. We're only able to identify a nearest neighbor that shares drug sensitivity, immediate nearest neighbor, about a fifth of the time. But we're really hoping, and obviously this goes up as we're including more and more neighbors inclusively, but we're really excited to see where this space of genomic similarity continues to evolve in the field. So kind of a method summary, the Molecular Oncology Almanac is a clinical interpretation method and database to evaluate individual genomic profiles for precision oncology utilizing capture and transcriptome sequencing. This method aims to serve as a decision aid, enabling rapid characterization of molecular profiles by molecular pathologists, physician scientists, or researchers. All provided data is viewable in the outputs, no data is hidden. This is really important to our molecular pathologist partners. And as I mentioned, all the code is available on GitHub under open and permissive licenses with release notes, documentation, and our standing operating procedure. And again, I also mentioned that we wanted everything to be packaged, so it does not require an active internet connection. The Docker, and you can, if you download it, everything is packaged with the software. We want to be sure we're adding value, so we'd love to collaborate. But before I move on, any questions or comments about the method?
I have one question, and you might still be getting to this, but you talked about expert curation in your figure with your pipeline. Yep. And I know that you guys offer like several interesting ways of kind of adding content to Molecular Oncology Almanac. I was wondering if you're going to be covering any of that in this section of the talk or later.
Yeah, actually, so I end up choosing to leave that out, but maybe I should have added it for this audience. Thanks for bringing that up, Alex. That's an excellent question. So I don't have slides on it, but we added several different ways for folks to contribute to the Molecular Oncology Almanac. So one is on our website, we do have a way for you just to submit a form to recommend a paper and a biomarker. This is directly inspired by the really fabulous work folks have done in Civic to really promote crowdsourced clinical interpretation. I remember presenting at conferences early on, and the most common question I received is, what's the gold standard for clinical interpretation? And there is no gold standard because the landscape is always changing. There's new papers coming out all the time. And there's even been meta-studies, both from the MetaKB and other ones. And overwhelmingly, the consensus is that these databases generally doing pretty darn good with consensus regarding FDA approvals and clinical guidelines. But the scientific literature out there for beyond guidelines is just so vast, and all these different databases are different scoped, that we really need to be working together to kind of put this kind of collectively harmonize and represent the academic literature in a structured way to be shared. So again, crowdsourcing is a fantastic way to help with that, as well as data harmonization. So we have the form on the website. We also made a Google Chrome extension, which has a 5-star review, I must say, where folks, if you're reading a paper and have the Chrome extension installed, you can kind of click, send this paper to the folks at Dana-Farber, and it'll fire off to us, and we'll see that. Then we can curate, like do the additional standardization and then add it to the database. You can also open a pull request on GitHub, email us. I think, did I miss anything, Alex? I think the main things were the crowdsourcing form and the Google Chrome extension as kind of ways to hopefully reduce friction for crowdsourcing.
I think all of that reflects what I kind of skimmed on the SOP for MOAlmanac. And I just, I think it's great that there are multiple ways for the community to contribute for people with different levels of expertise. I know Andreas had his hand up patiently for a while. So.
Thank you for the question.
I have a kind of follow-up question on the other end. Say you said there is an expert revision of the. So I understand here you describe many ways to suggest new interpretation. How do you handle, or how do you say your team or your clinical collaborators handle the volume to decide to review and add one interpretation to the knowledge base?
Yeah, no, that's a great question. And so thank you, Andrea. The question is really, as Civic and OncoKB and all these knowledge bases have, how do you decide what, like it's a fire hose of new academic literature coming out, how do we decide what to work with? And it's really targeted. And so rather, frankly, we do have a giant folder that's private on GitHub of just papers to be reading that we grabbed off Twitter and just as the journals came up with. And our strategy is really to kind of, instead of trying to ingest the whole fire hose, to more partner with groups intentionally to say, what do you recommend we're missing? We have these, we kind of have a structured reference template when we meet with collaborators to kind of go through their cancer ontology of interest for them to look at it, see what we're missing, see if there's any important papers we're missing, any conferences that are coming up that they think we should be looking at. But like, for example, we're working with the osteosarcoma and leiomyosarcoma Count Me In project. And part of that was working with them for 6 months, curating the literature in that domain and seeing what's relevant in that cancer type. We're also kind of have an ongoing collaboration with some folks in the United Kingdom for brain cancers. We're kind of similarly kind of looking at the literature there. And so that's the strategy we have taken. I don't know if there's a right strategy, but it's something we found to be a bit more manageable and intentional, especially with the more out there evidence, so to say. Another thing we do is that we try to be as explicit with the relationship as possible. So in addition to kind of therapeutic sensitivity, resistance, and disease prognosis, we also have some papers that are not sensitive or does not confer resistance. We try to make that extremely explicit in the algorithm and database, just so it's as clear as possible. But I think this is a global problem, and why I'm super excited about these harmonization efforts. I'll kind of get to it, but one of our roadmaps for this year is to kind of redo some of our underlying database and then add some more API endpoints. And one of the things we'd love to do is structure the API and add API endpoints that will restructure the genomic alterations for the VICC standard. So to allow further ingestion to like the MetaKB or maybe even additional ingestion into our algorithm from other databases. So I kind of want to answer a few different places, Andrea, but how did that answer your question?
No, that's great. Thanks.
Yeah, no, and I think it's an open problem. And again, there was a study in 2020 in JAMA Oncology, and I'm blanking on the author's last name, where they looked at 5 or 6 different of these knowledge bases. And again, what they found is that just no one database subsumes one another because the scientific literature is so vast and they all have different scopes. And it's really hard. and NLP is not quite there yet. I know there's a startup called Scite AI, I think, SCITE, where they're basically kind of doing a Reddit upvote, downvote way to do it, where you can type in a query and it'll bring up papers, and the model, it'll try to grab how many papers support this relationship and not support it. I think they're probably the most advanced with getting NLP to work. I'm also really excited about there's a group called Connected Papers where you can kind of see similar citations, and I think that'd be a really fabulous way to kind of help with this database curation as well. Cool. Great. So I'll move on to the next section. It is shorter. I think I did talk a bit fast at the beginning of the presentation. So in this next section, I'm just kind of going through some limitations of the methodology, as well as some future directions. And to start with, I think there's maybe 5 or 6 that I added to this talk. So one limitation is that we do not explicitly report oncogenicity. And this is a very intentional decision early on, just because there's so many different software approaches for characterizing oncogenicity in missense mutations. And it's, as this audience knows, it's a bit harder to do in the somatic setting than in germline, where kind of ClinVar is a really wonderful resource. But we kind of try to do this by proxy, and then kind of leave the evaluation up to the molecular pathologist or the reviewer by annotating with these several other data sources. But this paper came out actually from many people on this call, I think, where there's a recommendation from ClinGen, the ICC, and the ACMG now to always be reporting an oncogenicity classification in addition to the clinical interpretations. So we're really keen on kind of expanding out our software suite to be incorporating the principles from this study in the formal recommendations to be making a call for oncogenicity. And in truth, we are, of the data sources we have, a lot, like I think we're 75% of the way there already, because we have population frequency, we have ClinVar for germline, we have cancer hotspots. The one thing our tool did not ingest was kind of computational estimates of oncogenicity, which the paper recommends, and they make a few recommendations. So we're currently scoping what we think is the most thoughtful way to implement that. And I know some folks in this call have thoughts on that, and some other folks such as the Method-to-Worm portal and CGI, both in Europe, kind of have taken their own approaches to that. But I think the computational estimate of oncogenicity for missense mutations is kind of the most up in the air for the field. So something we're working on. Another limitation is that we do have some non-standard terms that we're working to eliminate in the algorithm. So for example, this is kind of more legacy terms that are still in the algorithm. We have predictive implication for kind of the evidence tier of the cataloged, of the matched record, and then bin for the genomic associations to different databases. And really, I mentioned we're going to be working our database this year, and part of that is to kind of smooth out some of this language, which is a bit unclear. So in this figure, you can kind of see in our reports, you can kind of click on predictive implication in the bins that form more of an explainer of how genomic alterations are matched to the database. So on the left, the evidence tier of the underlying catalog genomic association. And then, are you matching just by gene versus all the way to the variant consequence or protein change, for example. And for future direction, I mentioned we really want to simplify language and terms and kind of use more standard terms at this point. There's a really fabulous effort called commoncancertestingterms.org. And I saw them present at the participant engagement and cancer genome sequencing set of conference calls. And essentially what they did is they worked with, I think, 50 or some odd providers and organizations, and basically made a tally of, all these synonyms for cancer testing and reporting that I think we are all trained to use as researchers who are writing papers and don't want to be repeating ourselves all the time. But actually, when we're making these reports, all these synonyms can be really confusing from those uninitiated or patients. And they were able to kind of smush down these 50 some odd terms and getting feedback from patients, researchers, providers on what a recommended term may be. And they really settled on, for any somatic associations, referred to as biomarker testing. And then for inherited associations, be very clear that it is the genetic testing of an inherited mutation or inherited variant. And these things are somewhat obvious, but I think especially us in the academic setting, we're used to the more nuanced, like using nuanced terms and using different terms to not be repeating ourselves. And so we're going to be going through our nearly 900 associations in our database to kind of clean up the language, because the blurbs we have are written by the team. Then also cleaning up some of the terms in the API and the reports and such. And to that end, another limitation is that our reporting at the moment is only for the clinical associations at the moment. So again, our reports are less than 1 megabyte, entirely self-contained interactive HTML report. But a future direction would be to revise these reports to maybe have additional ones. We're not for technical details. We're not sure, maybe we should be making reports, separate reports for patients versus providers versus researchers. These are open questions for the field. In the autumn, I read 30 or so papers, just kind of about the return of results. And I was actually really surprised that there was only 2 papers that described kind of A/B testing of the return of results in reports. And one of them was a for-profit company that was only describing their process of going about the A/B testing. They didn't want to share the actual findings, because they're a for-profit company and want to have their reports be kind of a competitive advantage. But this is something we're really interested in. There's ongoing work to try to define the kind of minimal and optimal criteria for reporting, but it's open problem in the field, and it's something we're thinking about a lot, and it's something I'm personally really interested in. So I'm excited to see where the field goes, and I'm excited to keep iterating with partners on designing better reports for patients, providers, researchers, the whole gambit of users. And this figure is from a technical report from Deep Variant, which Google did a fabulous job with, on. And lastly, as I mentioned, we're only ingesting the MOAlmanac for our underlying database for the interpretation in the algorithm. So future direction would be to incorporate additional standards and hopefully work with you all about maybe adding additional algorithms into databases into the algorithm that can be deployed and fully containerized. For our partners who cannot be connected to the API, we're working with folks in Veterans Affairs and several other partners that don't, like CLIA labs that can't use an active internet connection. We're working on adding endpoints to our database to support the VICC output for variant representation. And yeah, so we're excited to work with you all on both the ins and the outs for the database and algorithm. So thank you all very much. Really appreciate being invited here. Thank you, Alex, for the invitation. We're also going to be hosting a workshop at this year's CGC conference. So I'm really excited to see all you there because I wasn't able to physically come last year. So I did a recorded talk, but really excited for this year's event. We'll have a workshop, looking forward to the hackathon, and looking forward to all the talks and conversations between. So thank you all very much. Yeah, happy to take any questions or comments. Stop sharing. Great.
That was an awesome presentation. Thanks, Brendan. Any questions from the group?
Hi, Brendan. I have a question. Thanks for the presentation. How frequently are the interpretations from practice guidelines, drug labels, and clinical trials updated in MOAlmanac?
Yeah, great question. So we shoot for a database release every month. The goal is to have something out the first Thursday of every month. Lately, we've been mostly focusing on FDA approvals, and we're scoping right now. Actually, this month we're revisiting the NCCN guidelines and starting with the lung cancer associations. So we kind of did a batched one in 2018, 2019, but then with COVID, like we weren't able to meet with as many people as much, and so rather than kind of having a updated like NCCN guidelines the whole time, we kind of have a versioned version, but shoot for a database update every month. We've been extremely good about keeping up with the FDA approvals, but the guidelines did get a bit lag behind because of COVID. And then, as I mentioned, because we switched to kind of trying to read in as many papers as possible to the more targeted approach of partnering with people, do the database curation, adding those, it is a bit more sparse. Maybe like once a quarter, we're adding additional academic literature for the database update, but you can go and go to our website. You can check out the release notes for most nearly every month. Thank you.
Thank you for the answer.
Thank you for asking.
I have another question actually. Have there been any initiatives to compare the clinical interpretations from Molecular Almanac with other knowledge bases?
Yeah, so we did one in this study that I mentioned that came out. In particular, we looked at comparisons to OncoKB and CIVIC, and we also looked at comparisons to a couple of clinical trials of the recommendations they made for the patients in the clinical trials prospectively versus what we are annotating and evaluating retrospectively. In the first case, as I mentioned, overwhelmingly, there's agreement between all these knowledge bases on the FDA approvals and clinical guidelines. But the more out there evidence that's sourced from papers, there's less concordance. I forget the exact number off the top of my head. I have some slides I can send you at a time. We're also included in the supplementary of the study. But just with the more out there evidence in the scientific literature, it is a bit of a fire hose. So kind of every database is scoped a bit differently. And they're grabbing different papers for different reasons. But then with the comparison to clinical trials, again, we did really well with anything associated with a FDA approval and biomarker clinical guideline. But if the molecular tumor board in the clinical trial did something that was either compassionate access or kind of a one-off thing, that tended to, we got, I think it was like 40% concordance with that. I'd have to look at the paper to get the exact number though. And I think that's representative of a larger theme of what makes this field so challenging and why it's so wonderful. We have so many harmonization efforts to try to collectively work together on this.
Thanks.
That answers your question. I know it was a bit more of a nuanced answer.
Thank you for the answer.
Thank you for asking.
Can I ask about how you guys manage, you talked about NCCN guidelines and trying to somewhat stay up to date with them. How do you deal with their fairly restrictive license and copyright terms?
Yeah, so we
how do you approach that?
We reached out to them ages ago, and the way we're managing it is like everything's citation driven. So we're not like we're citing it. We're not like, it's not in the algorithm, so to say. I'd have to look back at the email to get the exact legal language. But we got the okay to cite what they're using. So we're not including their, like, we're not publicly sharing their slides or anything like that, but we're just citing it. That's a really important question, so thank you for asking, Obi.
Sounds like you guys came up with the same solution as us, where it's sort of like the most you can do is kind of point people in the direction of an appropriate guideline, but not much more.
Yeah, no, exactly. And I skimmed over it a little bit, but in the reports, we have it so you can click on the source is always directly there, and in the report after the blurb there's a little hyperlink to kind of click, and it will bring you as far as you can to the citation. And going back to the beginning of the presentation about, you know, one can imagine a future of kind of a, you know, a super awesome interpretation system where it's running the whole clinical basket trial on itself and making the recommendations and actually giving the patients whatever therapy comes up highest. We're not going for that future. We are really trying to just help with the knowledge acquisition and sample contextualization of these individual patients, because there's so much information out there, and it's a lot for providers and molecular boards to stay on top of, especially when you consider access in different regions. In the United States, we're really lucky where the FDA and NCCN and such are kind of providing federal overall approvals of guidelines, but some collaborators in Europe, the issue is that the ESMO and EMA are providing approvals for the EU, but then there's also a second layer of approvals that are done nationally. And so like, I know some partners in Ireland, they are, getting foundation sequencing on their patients, but the approvals like aren't relevant to that setting whatsoever. So it's not just like a volume of information, it's also that the field needs to cope with. It's also being able to make systems that will tailor the knowledge to a specific area. Did that answer your question, Obi?
Yep. No, that's really great context. Thanks.
Oh, thank you. Ian, you have your hand up.
Yeah, hi, this is Ian King from University Health Network in Toronto. Your comment just now about Ireland and different jurisdictions really rings true for us. An FDA approval is is a pretty good sign that it's going to be useful to somebody, but these things differ. I would say for Canada, if the payer thing is the big thing. There's a definite sort of gap between, like you put it, the long tail of actionability and the ability of people to pay for things. And what I wonder is are you trying to link this to clinical trials available at partners in any way? The thought that we had was that it might be possible to have a catalog of all the clinical trials available at Princess Margaret, where I work. I think this probably exists, but that's a very difficult thing to actually get a handle on. Someone's probably keeping a record of that, but we don't really know who and how. So I'm just wondering if you've thought about that, because we definitely find that we recommend all kinds of great things to people, and there's just no way that they can get access to those drugs. And conversely, people have access to stuff that we don't know about, and actually, things that aren't in the literature prominently are represented in trials that our oncologists care a lot about. So I'm just wondering if you had any thoughts about that, about matching clinical trials?
Yeah, no, exactly. That's a great question. And I emphasize and reiterate all of the points you mentioned about access and affordability and insurance payers and such. It's another layer of complexity that makes this whole field, makes it really challenging for everyone. Oh my God. I'm so sorry, Ian, I just totally blanked.
It was really two questions. It was actually more of a comment that that the money aspect of it is actually quite huge, because these drugs are all super expensive. And just wondering if you thought about the trials matching, because it's kind of one of the logical next steps is to actually help people find what's really available on the ground. And for a particular institute, I'm sure Partners has pretty much everything that there is. So
yeah. Thank you. I apologize. This is something we've thought about a lot. There's a project out of Dana-Farber, actually, from Ethan Sarami's group called MatchMiner that you might have heard of. They published a couple years ago, and basically they designed a standard, and they called it clinical trial markdown, where it's a big problem with making these clinical trial enrollment criteria programmatically accessible, so we can have these programmatically accessible clinical trials that we can then programmatically match to the trials with. And they've done a really fabulous job integrating this into Dana-Farber systems. And they also just had a tumor board paper come out last year where they're actually talking about MatchMiner matches 2 active patients at Dana-Farber. Super exciting. I know one challenge that they have is that if you have multiple trials being matched to, how do you, it's really up to the molecular pathologist or an oncologist to decide which trial is most appropriate, but these clinical interpretation systems and knowledge bases might be able to help, or can help them figure out what trial might be best for a given patient. And that's something we're interested in connecting the pipes on, and are kind of an active conversation and collaboration about. But to your point, I think in their MatchMiner paper, I think they kind of undersold themselves a bit, because all of that, again, as with the curation of literature that we have in this space, all of those clinical trials were manually entered and manually standardized, and it is a tremendous time-intensive activity. And I know they have active people working on it to try to keep up with what's available at Dana-Farber. But getting that Princess Margaret, I can send you Ethan's email, but I bet they'd love to kind of talk and learn more about how it can help folks at Princess Margaret. But
that would be, it sounds like exactly the solution that that one, and yeah, as far as manual curation is actually kind of the only way, because clinicaltrials.gov isn't structured in that way, and it has to be done manually. So we experimented with the IBM Watson system when that existed, and that was supposedly using natural language processing to do that task, and it probably actually did a pretty good job. I mean, we have no idea to the extent to which NLP was actually used, but it probably did a pretty good job of that too. So I wonder if anybody is attacking that. But it sounds like, but that's exactly, it's exactly what I'm talking about, and that sounds like a really exciting MatchMinder. Sounds like a really exciting thing. So
yeah, check out. I'll look up your email and shoot over to you after this call as well.
Thank you.
Yeah, so I know we are at the hour. I know folks have back-to-back meetings and such, and I know some folks are AACR. So thank you all for being here, and I'll hand it over to Alex to do proper outros.
Thanks again, Brendan, for your presentation and that great Q&A. For those of you that are interested in Brendan's workshop, he did mention he'll be presenting kind of a clinical getting started workshop at for the MOAlmanac at the CGC annual conference. And of course, he and many others on this call will be attending or participating in the VICC Civic CGC or ClinJit Somatic Hackathon ahead of CGC. So we hope to see all of you there. If you have any follow-up questions, feel free to send around an email to Brendan. And we'll see you all on May 2nd for our next VICC General Call Series. Thanks, everyone. See you then.
Thank you.
Thank you.